Okay, in the last slide, there was a malfunction, so I'm just going to go back over and then add to the checkpoints. Again, we said the first checkpoint is at G1, and that checkpoint is going to allow us to check the DNA to everything that you've been exposed to. So here's that checkpoint that we're referring to. Here's my little stop sign. So it's going to, again, check existing DNA for mutations. So everything that you've drank, smoked, any radiation that you've been exposed to, whether it was sun or going to a facility for an x-ray, etc. So it says it's going to check the DNA to make sure there's no mutations. But if there are mutations, it says the first thing that we need to do is try to repair it. So we're going to try to repair. If we cannot repair that DNA, then the next thing that we want to do is to send that cell into apoptosis if the repairs were not possible. Okay, then we go into checkpoint number 2. Checkpoint number 2 occurs after we've replicated the DNA. S stands for synthesis. Synthesis of what? Synthesis of DNA. We're replicating our DNA. So in G2, it says we need to check who? Newly replicated DNA for mutations. It says if a mutation exists, number 2, it says we want to do what? We want to try to repair it. Now, if we cannot repair it, again, we want to do what? Send this cell into apoptosis. And if you remember, apoptosis means what? Programmed cell death. Last one, M phase, but specifically in metaphase. If you remember, the 5 stages were prophase, prometaphase, metaphase, anaphase, telophase. Well, we're looking specifically at M phase, but specifically at metaphase. Why? Because what happens in metaphase? In metaphase, the chromosomes align in the center of the cell. So what do we want to do? Erase that or scratch it out. It says, well, what are we checking? It says, 1, we are checking to, I don't know what's going on with this pen, to see if chromosomes are aligned in the center of the cell. Why? Because this ensures equal, oh mercy, division of the chromosome material. So we want to make sure that the chromosomes are lined up in the center, because if they are not aligned in the center of the cell, okay, when the centrioles are pulling on these chromosomes, it says that when the DNA separates at the centromere, most likely when this one separates, we could have one of the cells that ends up with what? So I'm going to choose this, when this guy divides, okay, that this cell has this chromosome, that chromosome, but because this one wasn't aligned in the center of the cell, we ended up with too much DNA material. So when they separate, segregate, move into different cells, one of the cells is going to end up with too many. If we went all the way back to our first slide, so you make sure you do this. I'm not going to try to do it, but if you looked at chromosome number 21, it said that the one of those individuals had 3 chromosomes. But remember, you always get 1 chromosome from mom, 1 chromosome from dad. But if non-disjunction happens because these guys weren't aligned in the center of the cell, somebody might end up with what? Too much DNA. Instead of you getting 2, you ended up with 3, because you get 2 from this one, and then when the other cell had the other extra one, then you're going to end up with 3 chromosomes. And since we're talking about Down syndrome, 3 chromosome number 21. All right, so these are checkpoints. Most likely, this could end up being an essay question. But again, okay, so the number 2 to this is what? Same old, same old. And I've ran out of room, so I'm not going to write it. It says, if mutations exist, then we want to do what? We want to try to repair it. We want to try to line this guy up. Try to get all the DNA in the center of the cell. It says, if we cannot get it aligned in the center of the cell, and of course, since this has happened, we do know that some things do get away. It says, we want to try to send this cell into apoptosis, but I guess everything doesn't always work as perfectly as it is designed to work. Okay. All right, so now starts talking about the phases. G1. G1 stands for, we said, growth. All right, so in G1, we want to talk about the cytoplasm doubling in size so that this new cell is going to have the right amount of cytoplasm, and as well as all of the organelles. Each cell is going to need a copy of the organelles that were in the original cell. A minute ago, we just talked about this. S stands for replication of the DNA, so synthesis of the DNA molecules. And in fact, there's billions of bases that have to be copied exactly. Okay? And then the final growth in size, make sure we got enough cytoplasm, etc., etc. So our final preparations for division. Okay? So here we go. G1, then moving to S, then G2, and then finally mitosis. Okay, so let's move into mitosis. Prophase is going to be your first recognizable stage of mitosis. One of the things, if you're looking at this, which is the animal cell, you can tell by the shape, and that is that you can see the nuclear membrane breaking down. You see that this nuclear material seems very dark. Why? It's because this is when the chromosomes will condense, or the chromatin net will condense into chromosomes. You don't see the nucleolus, but there's a nucleolus there that's going to disappear along with the nuclear membrane. And then also the cytoskeleton has to break down, and that material again is going to be used to form, those lovely spindle fibers. And then one of the other things that's going to happen, remember you have the microtubule organizing center for the cell. It says those are going to move to the opposite poles of the cell so they can start shifting around the chromosomes. Then in prometaphase, it says in prometaphase, we begin to develop these entities that are called kinetochores. So it says the kinetochores develop and they attach themselves in the centromere region. Remember, the centromere is what holds 2 chromatids together. where it says those kinetochores will attach there, so that once we've got our centrioles, says they have a point of attachment for the spindle fibers. Okay, so these are protein cores, that complexes that develop and attach themselves to the centromere region of each chromosome. Okay, then metaphase. So now we've got an ability to move the chromosomes about the cell. So in metaphase, we want to move them, if you remember from our checkpoint, it says why. We want them right in the center of the cell. We want them lined at that equatorial plane Y to ensure equal division of the chromosome material. We want to make sure each cell has exactly half of whatever, an identical copy of whatever the original cell had. So we want to make a copy of it, and then make sure that each cell gets an identical copy of everything that was there. So if you started with 4 to 6 chromosomes, you want to make sure you put 4 to 6 chromosomes back in each cell. All right, then in anaphase, it says we're going to pull those sister chromatids apart. So you can see that the chromatids are being pulled to the opposite poles of the cell. So here's those centrosomes, and they're pulling those chromosomes to opposite poles of the cell. So the sister chromatids, where they're going to separate at the centromere region. And again, they're going to be pulled to the opposite poles of the cell. The other thing that we can see that's going to be happening is the cell is going to start to pinch in two, so that cytokinesis is going to actually begin at the close of anaphase. Wonderful. Okay, then finally telophase. Telophase, we can see that those actin filaments are causing a constriction between the cells, so that they eventually will pinch into two. Now, one other big thing that we want to say about telophase is the opposite of prophase. So everything that went away in prophase is going to come back. Everything that came in prophase has to disappear. Okay? So the chromosomes begin to uncoil and form the chromatin net again. It says the nuclear membranes that went away. Now we're going to have 2, because 1 in each new cell, but the nuclear membranes are going to form around those daughter chromosomes. It says the spindle fibers that were forming, says they have to go away. We don't need them anymore. They're going to break down and become the cytoskeleton. And then it says, of course, cytokinesis is going to occur. What is cytokinesis? If we're talking about in an animal cell, we're talking about a cleavage furrow actually being formed, and we said that cleavage furrow would be formed from actin filaments, which cause constriction between the two cells, which eventually will cause them to pinch into 2 cells. And then what we want to look at is 2 definitions, karyokinesis and cytokinesis. Karyokinesis, again, is the division of the nuclear material. We know when that started, when that nuclear membrane started to break down. So in prophase, it ended in anaphase. Why? Because that's when we separated the 2 sister chromatids. Okay, and then we'll look at cytokinesis. Cytokinesis is a division of the cytoplasm. So we know that began when? At the close of anaphase, and then we know that it takes all the way through telophase. They say, but it doesn't reach a completion until interphase starts. So to go back over these stages again, one of the biggest things you can remember about mitosis is that it produces 2 identical daughter cells. So let's run. I've got one other thing that I need to do, and that's plant versus animal cell mitosis. Instead of us forming a cleavage furrow and plant cells, we're going to form a cell plate and not the cleavage furrow. And that cell plate is going to come from Golgi vesicles, which fuse to form these membranes. Okay, and then later they'll bring along cellulose in order to strengthen the wall. So cellulose will be delivered later in order to strengthen the wall. Okay, so there's mitosis. That's all of the stages. And you see us pulling those chromosomes away, and then the cell actually dividing inside of kinesis.